This is podcast number 41. So, continuing our review in these last 2 weeks of the semester, I want to remind you that your recitation 14 is due on December 4th. You want to get together with your group and complete that as a group. It's worth 10 points, but you can earn up to 14 points. And you have just 15 minutes to do that. There's no extra material. It's all a review just in that quiz. And I also want to remind you that the final is on the 7th, Wednesday of finals week. It's at a different time than our other tests have been, 12:15 to 2:15. You're going to use Respondus like you have for all the other tests. The final is worth 25% of your grade. It'll have 75 multiple choice questions on it, 9 extended response questions, of which you pick 5 to answer. And just because something is or is not in this review this week doesn't mean anything. It's not a clue. It doesn't mean this is all you have to study. This is just to remind you of the things that we talked about this semester. Okay. We talked about the difference between channels and pumps. And channels are moving things through the membrane by diffusion. So if it is higher concentration outside the membrane, then it'll come into the cell. If it's higher concentration in the cell, then it'll leave the cell. And those are channels. Whereas pumps use ATP to move things across the membrane against the concentration gradient. And we talked about specifically the sodium-potassium ATPase as an example of a pump. We talked about the special case of diffusion of water through cells, through cell membranes. If the cell is sitting in a hypertonic solution, the cell will lose water to the solution and the cell will shrink. If the cell is sitting in a hypotonic solution, the cell will gain water from the solution and the cell will swell. Depending on what kind of cell it is, it'll swell to the point of bursting if it's an animal cell. If it's a plant cell, it will just become stiff because the plant cells have a rigid cell wall on the outside of the cell membrane. And isotonic solutions have water moving in and out of the cell at roughly equivalent rates. So it's not that water's not moving, it's just that whatever water moves in, water moves out at the same rate, and so the cell will stay relatively the same shape. As an example of a pump, we talked about the sodium-potassium ATPase, how this changes conformation as a result of going through a phosphorylation cycle, and it creates a high affinity sodium site when it's facing the inside of the cell, and then that changes to a low affinity sodium site when it's facing the outside of the cell and the sodium leaves the cell to where there's already high sodium outside the cell. So this is against its concentration gradient. At the same time, it creates a high affinity potassium binding site and moves that potassium by releasing that phosphate into the cell where there's already high potassium inside the cell. So if you remember the word kin, K-I-N, K is potassium. Potassium is high inside the cell. Sodium is the other one. Sodium is high outside the cell at rest. And that is maintained by splitting ATP and this pumping sodium against this concentration gradient and also pumping potassium against this concentration gradient. Okay, here's an H5P question for you. All right, we talked about the idea of some organelles once having an evolutionary history of being free-living prokaryotes, and those were endocytosed or engulfed by another cell, and instead of being digested, they were instead kept for their beneficial properties. There's good evidence for this for modern day mitochondria in eukaryotic cells and modern day chloroplasts in eukaryotic cells. Mitochondria and chloroplasts have their own DNA separate from nuclear DNA. That DNA looks like prokaryotic DNA because it's circular and it doesn't have introns, just like prokaryotic DNA. That DNA can replicate independent of the cell that it's in, and there are multiple membrane systems around mitochondria, 2, and around chloroplasts, 3, indicating that they have been through endocytosis. We talked about the endomembrane system, the idea of a default transport pathway from the rough endoplasmic reticulum to the cis Golgi, through the Golgi stack to the trans Golgi, and from the trans Golgi to the cell membrane to release proteins that were made originally on the rough ER. We talked about different kinds of filaments that make up the cytoskeleton. Microtubules are the biggest diameter filaments. They are made of tubulin. Microfilaments are the lowest, the smallest diameter filaments. Those are made up of G-actin subunits, and intermediate filaments are intermediate between the biggest and the smallest diameter. They're made up of all kinds of different unit proteins like desmin, fibrin, keratin, etc. We talked about, again, this default pathway coming from the trans Golgi to the cell membrane, and via endocytosis, how the cell can take in material from outside the cell, bring it into the cell, have that fuse with either a lysosome or the cis Golgi in what's called retrograde transport. So this is anterograde in this direction and retrograde in this direction. We talked about specific cases where specific molecules are brought into the cell. For example, cholesterol. There would be a specific receptor on the cell surface. That receptor would bind a specific substance like a low-density lipoprotein particle from the outside cell fluid like blood. That would cause the receptor to change shape. It would cause that to be collected in a coated pit. That coated pit is coated with a protein called clathrin. That clathrin will polymerize around that pit, helping to form a vesicle sort of shape. The last little bit of that vesicle is cut off by a protein called dynamin. Then that coated vesicle enters the cell. It becomes uncoated, and the clathrin is recycled to do that again. This uncoated vesicle fuses with a compartment that has a low pH called an early endosome, which becomes a late endosome. And that low pH will cause the ligand that came in to uncouple from the receptor, and then the ligand can be digested in a late endosome or lysosome, and that's how the cell can bring in things like cholesterol into the cell. We talked about the dynamics of microtubules as part of the cytoskeletal network. Microtubules have a unitary structure of a protein called tubulin. Tubulin binds GTP, which can be hydrolyzed to GDP. The microtubules have a net addition end and a net subtraction end. It's not that you can't add things onto this end or subtract things from this end, just this end tends to be growing and this end tends to be shrinking. That has to do with the state of GTP, and on the positive end it's more likely that you will find GTP bound to tubulin than on the negative end. Okay, an H5P question for you. We talked about microfilaments. Microfilaments are made of actin subunits. Each individual actin subunit is called G-actin or globular actin. They assemble into these filaments, and this is just a space-filling model. These also have polarity in that they have a plus n and a minus n, and a net addition n on the plus n, and a net subtraction n on the minus n. There is an assay that you can do in a lab where you add myosin heads to the actin end, or to the actin molecules, and they will form this arrowhead sort of structure here. Let's see if I can draw this. So an arrowhead like this. Okay. And even though it doesn't look like this inside the cell, you can tell what the orientation of these actin filaments are, is because the pointy end is the minus end. And there's a mistake on this slide that's provided by the book. This should be the minus end. The barbed end, the wide end of the arrowhead is positive, but the pointed end is negative. And then intermediate filaments are intermediate in diameter. Lots of different kinds of proteins make intermediate filaments, and unlike microfilaments or microtubules, they don't have a plus n and a minus n, so they're not polar. Partly for that reason, they are very stable, but they can polymerize and depolymerize, and they seem to be more cell-specific in that the types of microfilaments you would have in a liver cell would be different than the types of microfilaments that you would have in, say, a heart cell. Microtubules, because of their polarity, can and are used by cells to move things along very big cells and not so big cells using these motor proteins, kinesins and dyneins. This one is a kinesin that physically walk toward the plus end on microtubules. Dyneins walk toward the minus end, and they can carry cargo. And these cargo components can be big, like a whole piece of a mitochondrion. Okay. We talked about different metabolic pathways. We talked about examples of breaking down carbohydrates, glucose as an example, and we talked about glycolysis and all the intermediate steps, all the enzymes from those intermediate steps. We talked about the tricarboxylic acid cycle, also called the citric acid cycle, also called the Krebs cycle, as sort of a crossroads in the cytoplasm, in mitochondria, not in the cytoplasm. That elements of breaking down protein, elements of breaking down fat, elements of breaking down carbohydrate, all of that meets at the citric acid cycle. We talked about the electron transport chain on the mitochondrial inner membrane and how highly reduced electrons on electron carriers NADH and FADH2 enter the electron transport chain at different points. They lose energy as they're passed down that electron transport chain, and the energy that is lost is used to move protons against their concentration gradient, creating a proton gradient that is used to power the FO-F1 ATPase. As protons flow back down their gradient, F1 rotates and orients ADP and P together to make ATP. Oops. Here's an H5P question for you. We talked about the breakdown of fatty acids and beta oxidation, how the rate limiting step is just getting the fatty acid into the mitochondrion through the CPT system. We talked about how fatty acids are broken down 2 carbons in the carbon chain at a time. That's called the beta carbon in the acyl chain, and this is called beta oxidation from them. You will eventually create an acetyl-CoA molecule from these pathways. That acetyl-CoA molecule will go into the TCA cycle and complete this TCA cycle just like acetyl-CoA coming from glucose. We talked about briefly breakdown of proteins, how some of the amino acids can be directly converted to an intermediate in one of the other pathways we talked about, like pyruvate or acetyl-CoA. And for the other ones, we just didn't talk about them. Again, this is going to end up in the TCA cycle. You recognize acetyl-CoA here again from the TCA cycle. We talked about photosynthesis and how there are different types of light harvesting pigments. In photosynthesis, there's antenna pigments that pass their energy onto reaction center chlorophylls, and the reaction center chlorophylls actually kick out an electron to an electron chain. That electron transport chain looks very similar to the one that you see in mitochondria, and it's the same sort of idea. These high energy electrons are moved down an electron transport chain where they lose energy, and that energy loss is used to move protons from one side of a membrane to another, and that proton gradient that is created is used to create ATP. We talked about how the products of those light-dependent reactions are used in the Calvin cycle, which is a reducing cycle. It takes CO2 from the atmosphere and it adds it on to carbohydrate molecules to make 3 carbon intermediates that can be put together to make carbohydrates for the plant. And we talked about how this particular enzyme here, Ribisco, here, CC1, yes, right here, sorry, the one that is taking CO2 from the atmosphere, adding it on to RUBP. CC1 is an inefficient enzyme, and it's affected by the concentration of oxygen in the cell.